basics, and especially the live stream.
Oh, yes. Is it okay for you?
Of course.
Okay.
Yeah.
So I just see this option here.
Okay.
Yeah. Okay.
So then the outline will be to actually understand why it's important, considering how it was born, then some of the principles, and then examples from our lab. Recently, we are trying to include this approach in our trials. So you will see some of the, actually, we're going to compare the results. Okay, so first, I think it's very useful to start with this framework that I think we discussed before, and it's the idea of translating the knowledge right from the evidence generation to evidence-based healthcare. And at the end, this process is basically our main mission, right? We are generating evidence with trials, with observational studies, and it's actually passing through a process of synthesis to finally produce a guideline, and this guideline to be implemented in the real world, right? The thing is, like, in order to do this, we actually are doing in the process inferential steps. We are going from information from a sample to a population, right? And this inferential process is actually following this classic Cartesian model, right? That probably you know, like it's basically the methodology of science, that you define a hypothesis, then from that population that you can test the hypothesis, you collect data from a sample. From this sample, you do some sort of initial data exploration, data analysis, and then with this data, you define probabilities, especially probability distributions, and then based on that, you do some inferences, draw some conclusions, usually in the way of testing hypotheses. So this process is always like iterative, and we repeat and repeat a lot of times. The only thing that is different is how can we conceptualize probability, the number 4, and therefore how we interpret the number 5. But the rest, any approach of that analysis is always the same. Okay, so now let's see that there are two different views of what is a probability, right? Like we could say there are two parallel universes of these definitions. One is the view of, I don't know if someone of you knows who is this person there? No one? I don't know. It's kind of famous actually, it's Sir Ronald Fisher. He actually basically started all the idea of the p-values, of the confidence intervals, and the F-test, the Fisher test, F-distributions, and a lot of things. And together with Neyman and Pearson, they created a process that is called the Neyman-Pearson-Fisher process. That is basically the thing that we do every day. That is, define your hypothesis, null hypothesis, then you get the data, and then you calculate test statistics, and you get a p-value, and you get a confidence interval in order to reject the null, right? I am assuming all of you know that, so I'm going to focus on the other side. Yeah. But the important definition is how this group define probability. And for them, it's based on frequencies, based on the frequency in the long term, on the long run for an event. For instance, the probability of raining will be the days that rain divided by the total days, right? So it's counting, it's the frequency of the event. And therefore, using this process, the inferences will be based on this assumption that all the inference depends on the likelihood of your observed data. And this data will be repeated randomly across different samples. So that is the concept. We get one sample and then we assume if we could repeat this 10 times, what would be the inference? You see, that is the approach that all of you know for sure. On the other side, oh, by the way, this approach is called Frequentist. So all the statistics we learn in PPCR, all the statistics of all our papers so far are using this approach, which is actually simple, computationally speaking, and it's also very objective in terms of you define cutoffs, you define specific assumptions that you can share with everyone in any part of the world. That's the advantage of this approach. On the other side, you have this guy that probably you know now is Bayes, right? And then this guy is actually older than the other. It's like 150 years before. the approach of Fisher and all this group. But there is a big problem with this, and was highly criticized at the time, because for him, the probabilities are subjective, are a degree of belief in an event. And therefore, you have a previous belief, and then you are updating this belief based on what you are seeing. So, for instance, the probability of raining will be the degree of belief that will rain. You see, so it's very abstract. It's really hard to prove, especially at that time. But in his mind, the inference process depends not only on the observed data, but also the unobserved data, the things that I could learn just by being human, by seeing stuff. You see, that's why you can imagine it was highly criticized, and nobody takes serious at that time, took serious at that time this idea. And that's why it was postponed until 1960s, 70s, 80s. You will see why, especially because of the computational power. But you understand why specifically now. So then, this is the summary, the mathematical summary of what he proposed. And the big advantage here is that you could go from a conditional probability, right? Like the probability of an event given another event, given B in this case, you could calculate using the inverse conditional probability. That was the beauty of this theorem. And we are using this for so many things, especially in engineering, to calculate and to optimize systems. But the thing is, like, it looks like it's not useful, right? If you see that, because at the end, why you just don't calculate the inverse, sampling the complement of the set, right? But sometimes it's not possible. Sometimes it's easier to calculate B given A than A given B. That's why it's very useful in some calculations. But that is not as important as this representation of the same theorem. So imagine like one event is a hypothesis. It's a specific statement, like for instance, that the treatment A is better than treatment B, right? This is our A, as our event A. So I call it their hypothesis. And then what we want is the probability of the hypothesis that we want given our observed data. Mm-hmm. So data will be the event B, right? So therefore, we're going to calculate, or we need to calculate the probability of the data given our hypothesis, if our hypothesis were true, would be true, multiplied by the probability of the hypothesis, divided by the probability of the total possibilities of data, what we call the marginal probability. So then, I know it seems strange, right? But let me go part by part so that you will understand why this is useful. So first, let's think, like, as I mentioned, the probability is subjective. So you have a prior belief, and then you're going to update your belief based on the data, right? So let's use the formula to do that. You see, now I put it with colors. So the probability of your hypothesis is your prior belief, So you are ready, for instance, I don't know, you had the clinical expertise and you saw that something is very frequent, or you run an observational study and you calculate the probability of the specific treatment is usually much better than the other treatment, or you have a meta-analysis published or a previous clinical trial published. That will be the probability, the green probability, that is named the prior distribution. Okay. Then the blue part is what we call the likelihood, and this is the same like in the frequentist approach. So we calculate the probability of our data given our hypothesis, and then we divide it by the probability of the total possibilities of the data. Here is the problem, because as you can see, let me just try to do a zoom here. As you can see, As you can see, in order to calculate the probability of the total data, you require to do a high dimensional integral. And that was the problem at that time, because nobody really could solve this by hand, especially if you have more than two groups. So then everyone forgot about this and they focused on the frequentist approach, okay? And then when you multiply the prior distribution by the likelihood divided by the probability of the data, you get the posterior distribution, which is the probability that represents how likely is your hypothesis given your observed data. Okay, so that is the important concept here. You are combining your previous belief with the likelihood of your data to get a posterior distribution that is telling you how possible is that your hypothesis is true compared to giving you data, observed data, okay? This is very different because in the frequentist, we never support or we never prove any hypothesis, right? We always reject the null or we fail to reject the null, right? That is the big change. And then at the end, this is just an example of how we calculate this total probability, that is the denominator, and we need to use a Markov chain simulation. That's why, as I mentioned in the past, nobody could really solve it until we get the new computers. So at the end, we can ignore the denominator. It's just to show you why this was complex before. But now that is easy. We do a simulation process to get random samples, like thousands and thousands of random samples, and then we calculate the total probability. But the numerator is the important part. So you get the likelihood from the data, you multiply by your prior distribution, and then you get the new probability of your parameter, in this case, the beta coefficient of a regression comparing group A versus group B, right? Okay, so that is the application of that. And let me give you another example that is very intuitive for all of you, I guess, is the idea of medical diagnosis, right? So usually we always start with a prior belief of the frequency of a disease, right? If something is frequency, if something is endemic, then because of your training, because of your experience, you have a probability in your mind of how frequent is, let's say, a specific infection, right? And then you request a test, then you perform I don't know, physical examination, or you request some blood test, and then you calculate, you know, the likelihood of this, because someone already calculate, right, which test is the sensitivity, specificity of the test and everything. And then what you are doing in your mind is actually a posterior probability after you update based on the likelihood, based on the test, multiplied by your prior belief. You see, and then you make your diagnosis based on this post-test probability, which is at the end a classic example of a posterior distribution. So at the end, we are always doing that. So let me do an example with this using the frequentist approach, right? In the frequentist approach, you always need to assume that the null is true. So you are assuming like the patient has no diagnosis,
sure,
right? And then you get a test, and based on the test, you reject the null. So, or you accept the null. So you will say, oh, I'm rejecting that the patient has no diagnosis. You see? And that's it. And that is not useful at all because you didn't get any conclusion, right? So then you don't know what to prescribe, what to do, right? So you see that the tricky part, like in the environments that we need to make a decision right away, we always use this approach.
sure.
It's in our minds, it's by default because our cognition works in that way. But for some reason in research, we use a null and do double negation to kind of advance failing or rejecting the null. That's the only way we use science. But it's counterintuitive. It's against our cognition, and that's why a lot of people get confused until now about what is the p-value and what is a confidence interval. You see, because they are thinking with a Bayesian mind. So this is just a quick example. So now let's do a comparison. I'm gonna present characteristics of the frequentist in one side and Bayesian in the other side to see similarities and differences and to understand how to interpret these differences. And then we're gonna see an example of one of our trials, and then we're gonna discuss how we interpret each of the components. So the first similarity is that the same methodology of science, like you always go from the population, you get a sample, and then you do a statistical test. And then you need to define a prior hypothesis. That could be that the mean difference under the treatment A is larger than the mean difference under the control, right? That is your alternative hypothesis, and then you don't need the null as the main hypothesis. You can focus in your alternative. That is the big advantage of the Bayesian analysis. But you will need to do the same. You will need to go from the population, the sample, and then do the test at the end. This is another very important difference. For the frequentist approach, your sample is just one alternative of infinite samples, right? But there is only one single true value. Let's say if we are interested in the mean difference between two groups, the frequentist assumes that there is one unique difference that is true. The only problem is that we don't have all the population to find this true value. Therefore, we select one part, and we assume that if we do thousands of new experiments, we could get close to this unique value.
Yeah.
But in the Bayesian approach, we assume like this effect estimate is not unique. It's a distribution. So you have different values of this that is changing depending on the scenario. So we are not obsessed to identify this true value, but actually see the probability of that this is happening. So we have a distribution of potential true events or true effect sizes. So that is the most important part, because therefore there is no need for a p-value, because the p-value and the confidence intervals are actually part of this idea. Like if we repeat and repeat thousands of times our same experiment, how extreme or how rare is the thing that we observe. And based on that, we reject the null, right? But if we don't need to do this thousands and thousands of times, we don't need the p-value, okay? So just as a reminder, the frequentists use 0.05 to reject the null, right? But we never can say we are finding evidence in favor of the alternative or proving the alternative. That is conceptually wrong. And this is just the definition of p-value. So it's the proportion of times of an observed event or more extreme event will occur in the series of repetitions, but only if the null is true. That is the most, sometimes the most complicated part to understand of the p-value. And in the other side, we are just calculating a probability that our hypothesis is true given our observed data. So we calculate a direct probability of A is larger or A is better than B. You see, not the null that A and B are equal. You see, that's that's why at the end is more intuitive and help us to phrase easily the conclusions. So this is, again, I'm assuming all of you know this. I hope so. So initially when you are doing the statistical test, you assume that the null is true, right? So then you put the mean or any parameter in the center based on your null, usually 0, right? Because you assume that there is no difference. And then you collect data, and then you see in this hypothetical distribution, you see where is your observed value. And if your observed value is on the extremes, right, then you will reject the null. That's the idea. But again, assuming that the null is true. Okay, that is clear for everyone, right? Okay, great. And then the other important aspect is, like, at the end we say 5%, but when we are using bilateral or two sides, actually it's half two. It's divided by 2, right? So then we need to usually include 0.025 in each side, because we care of more potential explanation, not only one side. Okay, great. So in the Bayesian methodology, we don't need to calculate this. What we calculate is the following: is a probability here, in the right side, is how probable is our hypothesis based on the posterior distribution of the parameter. Therefore, we assign a probability value. In this case, for instance, the probability was 0.89, right? So then that will be the most important part, not specifically the p-value in the frequentist. Okay, so we get a value that if it's closest to 1 is better. Okay, in the frequentist, if it's closest to 0, right? If it's going to a small and a small value, is better. But in the Bayesian is the opposite. And then The other big difference is that, oh, well, I already mentioned this part, skip it. The other big difference is this, the interval measures. In the frequentist, we use the confidence interval, right? How you interpret a confidence interval? Someone can tell us, just to be sure all of you understand that part. Any volunteer?
Yeah, like let's say if you do like, or you measure, I don't know, like certain amount of times, so that observation will be inside this interval, so like the confidence interval, right?
But what will be inside this interval?
Your like observation of what you're measuring, right?
You see, that's exactly a Bayesian mind.
The true answer, right?
Sorry?
The true answer will be inside the interval with 95% of confidence.
Exactly, is the true, it's not the observed.
Yes.
That's the trick. In the frequentist, we are always obsessed to try to identify this ideal true effect. So what we're saying there is like, if we repeat this 100 times,
It
right,
will
it will include 95 of these hypothetical trials will include the true value, you see? And then in 5, we're gonna miss. We're gonna fail to identify the true value. So, but actually exactly what you said, Jorge, is the credible interval, or the interval of credibility, which is the Bayesian counterpart of this idea of intervals. So there you could say this is the probability of observing my value. So my value will be between these two ranges, between this, in this case, 0.3 and 1.3. So if we take care of this distribution, we could actually see like the minimal effect that we could observe is 0.3. You see? So it's actually the 0.3 in favor of our hypothesis, our alternative hypothesis. It's not if we repeat 100 times the experiment. So at the end, usually they are very similar in terms of the centers, but when you are in the extremes, usually the Bayesian is a little bit more conservative, so are wider. So that's why it's really hard to do hacking, like p-hacking, or to really have a lot of false positives in the Bayesian methodology, because the intervals are wider and wider, most of the cases. So that is the, I don't know if that is clear, because this is very important to interpret our example. So that the credible interval, actually is telling you the possibilities of your value, of your estimate in your sample, in favor of your hypothesis. You see, so you can interpret directly as Jorge did. Like if you have these in different subsets, you could get from 0.3, the estimate, to 1.3, being your mean 0.8. Okay, and we're going to interpret from our...
Yeah, because I think that's important just to tell them here, guys, it's as Kevin showed, the Bayesians, and the Bayesian means what? It says, 95% probability.
Mhm.
And the frequentist, it means what? If you do it as 100 times, you see the difference. I think that you can think, because one is more concrete. If you do this experiment and you run, you get 95 times within the boundary. The other one is a probability. It's a more abstract concept, because you need to understand, and then what Kevin is also helping you is to understand how to calculate probability, but it's a more, so frequentists have more sort of concrete mindset, and the Bayesian uses information a bit more, as Kevin says, our brain is doing a lot of statistical analysis all the time. So when you wake up in the morning, you're going in there, and now I have the Apple, etc., the weather, it's much easier, but I think 10 years ago, so you go, that we didn't have all the devices. So yeah, it's fall, I'm going to Spalding, you look outside. People used to have a weather forecast outside of their home, the forecast thermometers, because 15, exactly. And now, so it's crazy. But, and then you imagine, okay, I'll go in fall, then I'll get a jacket, a overcoat, and that's it. So, but that's your brain thinking. Time of the year, this, and putting all the information and doing a prediction, okay? So, but I think that's the
Exactly,
beauty of
that's a
that.
different mindset. That's why
Yeah.
I'm trying to highlight this part because it's against our training.
Yeah.
Because we automatically try to be frequentists because of the PPCR actually and because of our training in med school, right? But at the end, it's very interesting to see the comparison. You will see when we analyze a real data. But the last thing, actually two more things about principles, okay? So the first principle was you will have probabilities. You will have probabilities in favor of your hypothesis, of your alternative hypothesis, and you're going to identify a credible interval of all the potential values, the true values that you can identify from your observed data, right? This is the credible interval in the Bayesian aspect. And then you will have the bias factor. This is another very important concept. This bias factor is going to divide these probabilities of your hypothesis, alternative hypothesis, and your null hypothesis to see if this evidence is actually in favor, it's supporting the hypothesis 1 or the hypothesis 0. And we have this scale that is kind of a convention in the field. So if you divide that and it's 1, then you will say you don't have any, you are not prone to favor any intervention. But if you start moving to 3, 10, 30, you will say that it's in favor to your alternative hypothesis. And if it's less than 1, it will be in favor to your null hypothesis. So the big question here is how much is big, right? So the convention tells that 10 or 30 is considered strong evidence in favor of your alternative hypothesis, right? It's kind of a similar concept of the Cohen D, right? The effect sizes, but in this case, you are comparing the two hypotheses. And we're going to calculate this from our trial. And then the last part is about the interpretation of these probabilities, right? Because how can we say what is considered strong probability or really decisive evidence? And there is different classification. This is one of the most accepted one. So if you have 0.75 to 0.95, you could say that is a positive probability. So you could say that your trial was in the area of positive results. If it's from 0.95 to 0.99, it's a strong evidence in favor of your alternative hypothesis. And if it's higher than 0.99, this is actually very, very strong. You will say it's a decisive evidence in favor of your active treatment. So those are different cutoffs, but the difference between this is not that fixed, like in the p-value 0.05, right? Here it depends, and you could argue that it depends on your prior knowledge as well. So that's why the one that is recommended to report is this Bayes factor, because this Bayes factor is actually representing the results from your data, comparing the two hypotheses. And then you can change the green part. The green part is your belief. You can change your belief, and you can have very skeptical beliefs. You could have really in favor beliefs based on meta-analysis or based on your hypothesis, and then you can do a sensitivity analysis using the Bayes factor. That is the beauty of this evidence, because you could use this Bayes factor to train other data sets as well, based on the previous studies. So here is just an example of the important role of the prior distribution. This is actually the big controversy, right? Because how can we know if our prior is the correct one, right? So, and the answer is we don't know. We need to make a decision. So that's why we have three main different types of priors. The non-informative, where you assume a uniform distribution. You could have a weakly informative, where your 95% of the density of your distribution has a lot of values, right? Or you could have a very informative that could have a specific range of values in favor of the active intervention or in favor of the control. So we could go from all of those and to see in which scenarios our data, which is the likelihood, the green one, change the conclusion, which is the posterior. You see, that is the beauty part, because then we could integrate this, for instance. We could get from our previous meta-analysis of trials testing the treatment A versus the treatment B, and to incorporate those in the prior. So then we could do the analysis with our new data to get an informative posterior distribution. For instance, in our TDCS studies or in the BNS studies, we could include effect size of previous meta-analysis on the same outcome to inform the conclusion as well. Or we could use what is called a exceptical prior, where we actually, in purpose, start with a prior that is against our active treatment to see if with our data, this could be shifted because it's so strong, the new results, that even with an exceptical prior, even though you get higher probabilities of improvement. You see, that is the flexibility that we gain with this framework. I don't know if that is clear or any question, because this is very important.
So Kevin, there is a question from our YouTube. So when we are using the pre-test probability and multiplying by the likelihood ratio in order to determine the post-test probability, and they say, are these considering the denominator of the Bayes theorem?
Yeah, we are considering all the time. The only thing is like to derivate that, we need to use a Monte Carlo simulation.
Oh,
so we solve the denominator so many times doing this process. Let me show you the simulation. Just to show you how it looks like. So in order to get the denominator, we need to do these Markov chains you can see there. So we are solving the equation like thousands of times with different denominators from a pool of random numbers. And then we repeat and repeat and repeat until we get new posterior probabilities one at a time. And then we complete the distribution density, as you can see there. And each line is a sequence of different iterations. So that is the thing that why before it was almost impossible to use this. But now we can do this in a few minutes. Like I did like 1 million iterations to get a really robust posterior distribution
for our data.
But of course, it requires usually R or Python. It's not completely in R, but it's not available in all the packages yet. Because first requires a lot of new understanding how to interpret the outputs, right? And requires a lot of computational power as well.
Maybe after one number, it doesn't change anymore.
No, because you start with a seed number, meaning you start always with the same random number. So you could replicate the iterations if you need it.
So after one number, it's going to be almost the same. So
change
a little bit.
All this Bayesian approach is based on uncertainty. Right? So it will be stochastic most of the time, but usually converges to the same point. So that's why at the end it's very robust. So going back to the principle of the,
and then we're reminding everyone. So then we did the Bayesian analysis without prior information in that case.
We assume uniformity. I'm going to show
you. I'm going to show you.
But okay, so just remember those cabals, those assumptions and conventions. Now let's see our data. So before that, just as, to summarize the potential uses of this. First, in clinical trials, because it's intuitive, so it will be really easy to communicate to clinicians, this treatment has 99% of improving your patient, right? Compared to say the odds ratio is this and the confidence interval is this and the p-value is 0.01, right? You need first to explain what is the p-value, what is the confidence interval, what is the odds ratio. You see, so that's the beauty. Just one number, if it's really in the extremes, it's really helpful. The problem is in the middle, as you will see. But okay, this is one. The second one is the possibility of sensitivity analysis, changing your prior. So you could say, for instance, a trial in the US has a really good effect, but then you could change the prior based on your setting and to adjust the effect based on your prior, or using different meta-analysis on different topics. So that is the interesting approach. You can get multiple ways to do the analysis, and if in most of them A is better than B, then you could say, yeah, of course, should be recommended A against B, right?
Yeah, and here it comes as well. I think that's why people get a little bit nervous, because this depends a lot on what you input. A lot of it's subjective, and it depends. So there's two factors here: the investigator using the site in the parameters, and also the reader being able to read, get a critical thinking of the Bayesian analysis. That's the challenge, because even for frequentist analysis, it's super hard, as you know, PPCR, to truly understand that and to truly have a critical thinking. I think that's one thing that is because you can change it, and I think that's a good thing, sensitivity analysis, because if we did all these scenarios, everything confirmed 99%, great.
You're fine.
Yeah.
The problem is like if you select the scenario where your drug is better than the placebo, that's the risk. That's
Exactly.
I agree with that. Yeah, that's problematic. That's why it's recommended now to report a set of specific sensitivity analysis
With a set of parameters. Sure.
exactly. Especially always including a skeptical one. Like the effect is 0 or even against your treatment.
Oh, I see, with the skeptical one.
Exactly.
Interesting. Sure.
So that is one alternative to be more fair.
Sure.
But it's not yet a standard procedure. Right. Then the other interesting part is the interim analysis, because you don't need to be Bayesian. You don't need to be a Bayesian trial to do Bayesian interim analysis to get a probability of the futility or efficacy with a lot of predictive power, even in small sample circumstances. So that's why now I would say this is the main use in the adaptive designs. All of them uses Bayesian approaches for the interim analysis, for the stopping rules and all that type of things. And then the last part is that we could optimize and especially improve the precision of the estimates because of the priors. So you will change in the prior. If your prior is so informative, you don't need that many patient approach, because you could do a sample size calculation based on Bayesian approaches.
Interesting.
And then you will get a sample size that is smaller. So it's recommended to put that in the sensitivity analysis in a trial application, for instance. What would be the maximum sample size under these scenarios? You see, which is, I think, very interesting. So the last two are very easy to integrate right away, because in our grants, for instance.
I see. Interesting.
As a sensitivity analysis, as an interim analysis.
Sure. Yeah, that'll be exactly. You're right. Interim analysis, a lot of them they use Bayesian analysis. Even sometimes they don't even spell out there is a Bayesian analysis, but it is. Yeah.
Exactly. Yeah. Exactly. And then I forgot to put it there, but it's also really, really important for meta-analysis, because then it's easy to integrate the previous trials to your new meta-analysis, especially when you're updating and updating.
A meta-analysis, yeah.
So it's a better way to do cumulative meta-analysis,
Sure.
But it's because you are always reporting the bias factor. So someone could use the progression of the bias factor across time instead of the effect size. So at the end, it's more transparent because you report the bias factor. Okay, so now let's do a quick example.
So question, so did they, you said the FDA has now a section on Bayesian analysis on their.
Yeah, it's recommended now.
Oh, it's recommended. You have some FDA-approved drugs with Bayesian analysis.
Yeah, actually, one of the vaccines recently for COVID was a Bayesian trial.
Nice.
And they actually stopped early because the probability of efficacy was so high that they just stopped it with 3,000 subjects instead of the 7,000 that they proposed.
Yeah.
So at the end, it's becoming part of the statistical thinking, but it's not to replace the frequentist. It's to complement.
Yes.
You know, it's not like we're saying, oh, we're going to change all what we learn now. It's more like to complement the conclusion. Okay. So any other question before we go to the example?
Questions, Newsom? Okay.
No? Okay. Great. So this is example that probably you know. It's our trial comparing PNS, transauricular vagus nerve stimulation, versus sham in healthy subjects. And we have, the 22 active versus 22 sham. So this is the frequentist analysis, right? We basically have the mean and the standard deviation pre in the active group, the post in the active group, same thing in the sham. And basically what we do is to do a regression where your factor is the groups, right? Your group is 1 is the active, 0 is the sham, and then you regress the changes in CPN, which is just the deltas. As you can see there, post minus pre, post minus pre. So you get a delta, and then you regress the delta using the group as a factor. That is the classic approach. And I have there the ITT because we impute in case we have missing data. So all the randomized were included. And then we have unadjusted and adjusted, because the unadjusted is just a regression I just mentioned. And then the adjusted includes also two factors that we predefine based on the table 1 to adjust for some potential imbalances. So just to have this extra precision. But at the end, the conclusion is the same. You can see that the beta 1 in this case is the difference between active and sham in the delta is 0.87 in the unadjusted and 0.80 in the adjusted, meaning the active group improves 0.80 points in CPM comparing to the sham. So, and this is almost 1 point of CPM, which is considered very big, actually, especially in healthy subjects. And then between parentheses, we have the confidence interval with 95% confidence interval, meaning we say, like, if we repeat, 100 times, right, the true value will be between these two 0.21 and 1.37. But remember, we are thinking on the true value, this in the population, if we treat everyone with active and sham. So at the end, what we learn is to see that the 0 is not there, right? To see that it's not plausible to have the null in your interval. And it's not, so we conclude that it's statistically significant, therefore the p-value is 0.007. So this, the proportion of observing this or something more extreme in the under the null, meaning the null is true, it will be really extreme, really rare, really in one of the sides of the distribution. And then we say, we reject the null, right? And then we kind of support the alternative, but not really. We just reject the null. And then we assume that it's potentially effective to modulate CPM. You see, that is the thinking in the frequentist approach that all of you know. It's just a quick summary. So, and this is the example, the graphical example of the differences, right? You have the sham. This is the deltas of CPM. You compare the sham versus the active, and then you compare the two distributions and you get a p-value. Then in this case, it's different because it's unadjusted. That's why 0.04. Okay, so any question about this frequentist approach? It's just a classic treatment effect regression. Okay, perfect. So now what would be the same analysis but in the Bayesian framework? So first, we need to define our prior, right? Our prior is not informative because we search in the literature, we discussed, it wasn't clear if CPM is changing in healthy subjects, only some studies using pain threshold. So we decided to be very conservative and to include a uniform distribution with almost zero information. Then we use these distributions, the active and the sham, and then we calculate two hypotheses. The alternative, which is active is better than sham, and the null, which is no different, right? So we have two hypotheses, therefore we have two likelihoods that we estimate from our distributions. And then we do a Monte Carlo simulation using the two of them to calculate the denominator of the Bayes formula, right? And then we get this formula thousands and thousands of times, and then we convert all this distribution in a unique posterior distribution of all our potential beta ones in the multiple regressions that we could do. And as you can see, everything is centralized to 0.8, going from 0.2 to 1.3, more or less. I think it's very small to see. But that is our probability, our posterior probability of the beta ones. Remember, the beta 1 is coming from the distribution that we are comparing the treatment to regress the delta of CPM. That's clear? Like, what is the beta 1 distribution? Is the coefficient of the treatment. So in the frequentist analysis,
remember it was 0.8 to 0.
Yeah, it's 0.8.
Yeah.
This is our beta 1 of this distribution, comparing, dependent is delta CPM, and the independent is a categorical, a binary variable that is group, let's call group, which has two levels. One is active and 0 is sham, right? Therefore, this coefficient is the difference between the group active and the group sham, right? So then we calculate the posterior distribution of all the potential beta ones using this non-informative prior.
Okay, 0.8 means that it's 0.8 better in active.
Yes.
Yes.
Exactly. Yeah. And then we obtain from these probabilities, we calculate the probabilities of our hypothesis. So we are actually subsetting the probability of being active being better than sham, right? So then I calculate only that part because that is what we really care about. So then this is the probabilities of active being better than sham when we compare the deltas of CPM. So as you can see, the probability is 99.999. So it's considered
To get a coefficient of 0.8.
Exactly, exactly. So it's really strong because at the end, in all these multiple simulations from the Monte Carlo, at the end, most of the times, always the active was better than sham.
Sure.
So that is super powerful to say in the paper. And the coefficient, it's very similar, 0.8, it's actually the same. And then the credible interval is going from 0.25 to 1.35. That means our parameter, in this case, the difference between groups, could go from 0.25 to 1.35. You see, now we can say that. In the interval, in the confidence interval, we can say that.
Yeah.
But a lot of people is doing it. That's the problem.
People, yeah, so sometimes they say confidence interval as the Bayesian analysis.
Exactly.
Credible interval. Yeah.
Exactly. And then I calculate the bias factor, which is 234. This is the probability of the alternative hypothesis and the null hypothesis.
Sure.
So as you remember, the convention is like if the bias factor is higher than 30, you have a strong evidence in favor to your hypothesis. So this is a really huge and strong result. The thing is like in those scenarios where you're in the extremes, right, it's not really different. The frequencies and the variation will be the same as you can see here, right? So first, any question about how we get to the probabilities or how we interpret that? So what would be the 99.99? The probability of
the shams better than
the
opposite, the active
of the
better than
active
sham.
is a probability of having better CPM rate of improvement in active compared to the sham.
Could we say that if we could say something about alpha, in this case would be 0.0001?
We could, but we don't have that concept in the Bayesian because the alpha is a significance under the null.
But
so we are not under the null. This is under our data.
But it's similar. In the frequency, the other analysis, you found alpha like 0.007.
Yeah,
this would be 0.0001
with a tendency to zero. Yeah, I mean at the end they are equivalent because they are extremes in the distribution, but they are conceptually different because this distribution is our posterior under our alternative hypothesis. Okay, the other ones under the null.
Under the null, yeah, exactly.
That's a big difference. Okay.
Kevin, can you just go back to this Monte Carlo simulation?
Sure.
I was lost in that part.
Okay. It's because I don't know if you were here when I explained the formula.
Yeah, I don't think so.
But the formula in the denominator, you have the total probability.
Okay.
And that total probability, the only way to solve it is to integrate the probability of A and to derivate the probability of A given B. And the problem is like if you have different factors, this will become a high dimensional integral. And then you will need to do like, I mean, it's possible, but it will take a while doing manually. So if you have more than two groups, then it will be multidimensional. And then sometimes you can just convert the derivation. So then it will be undetermined. And therefore, if you divide by undetermined, then you don't have any, right? So that's why we use a Monte Carlo simulation. We calculate one at a time. We obtain one value for the denominator one at a time. And we repeat and repeat this in thousands and thousands of times until we get a sample of the potential values. Exactly. And then we calculate that posterior distribution based on the Monte Carlo simulation. So it's a way to solve the equation, basically. It's just a tool. It's not to
that graph means, again, this is the each time you do it, it helps to determine that frequency, and that's a frequency distribution that you see it.
Exactly, of your beta one, of your parameter of interest. In this case, the beta one of the regression. Okay,
Kevin.
So, yeah.
So in the end, the Bayesian approach is a way to support a number found in with the frequency approach.
Could be. But
Right.
what happened if it's not?
To
Let me show you
support
one
or
example.
not, right?
Exactly. Let me show you one example. Actually, that's why I bring the next. This is a perfect example because you will report a trial. Actually, it's our trial. We're gonna say like in both ways, right? In both scenarios, the active is better than sham to improve
Let's see,
CPM.
maybe a drop. Maybe
But-
a drop, yeah, go ahead.
But what if it's not, right? Let me show you one example. So this is an example from a trial in the, actually in the trauma. Is it real or is true?
Yeah, I'll just.
Are you guys okay with it?
Yeah, if there is, there's like a fire alarm. Oh, stop now.
You're gonna an example that ordinary
Oh my
analysis
gosh.
doesn't show significance, but the Bayesian shows, right?
Exactly. That's the trick.
That's the
Then
game
what
changer,
you do-
right?
That's what you do, exactly. That's the,
That's the point, right?
exactly. But again, professor is coming. But let me explain you really quick what is this.
That's fine.
What's-
That's fine. I think it's just a test.
So this is an example where the people reanalyze a negative trial. Right? It was a trial that was pragmatic, therefore it was very heterogeneous with a lot of different things that you can't really control, right? But the evidence from those trials are really, really close to the real world, therefore are really valuable, like our PLP trial, right? So therefore what they did, let me show you here, they calculate, they compare two different approaches to provide blood transfusion with different components. But just let's say in that way. And they compared two groups, and what they found is that in the active, this is the results. They found that the relative risk, right, in the 24 hours was 0.75. So let's interpret that, what it means, 0.75. The first one is active, this is active, and this is sham. So 12.7 mortality rate versus 17 mortality rate. So therefore the relative risk is 0.75. So how do you interpret that?
There was a, yeah? Oh,
So it's 3/4, it's 75% of having the outcome.
it's protection, increased
That's
risk.
protection.
Yeah. And then the
75%.
condensed interval,
Where's this? Is this?
yeah,
Yeah,
0.2, 1.08.
yeah, but it crosses the one.
Yeah.
Yeah.
Exactly. That's the situation. So you have a point estimate that is telling you that the treatment A is protective, is reducing mortality at 24 hours, but the confidence interval is crossing one. Therefore, with our frequentist mind, we say it's not significant, right? But the
Right.
p-value is 0. It's marginal. It's not that good, of course. We would say that it's not at all. And then if we check at 30 days, it's the same story. The point estimate is 0.86, and the confidence interval again crosses one, right? So if we analyze this, of course, the main conclusion of this trial was that it's not significant.
Yep.
And in all the trials, they add, in all the guidelines to using this conservative approach versus the other approach, they actually recommend the first one, but putting a note, like based on the clinical expertise and recommendations, they would prefer the first one. That was and it was a huge controversy because the first one is very cheap and the second one was very expensive but safer. So it was like a lot of dispute in this scenario. That's why they reanalyzed this trial using the Bayesian methodology, and you can see what they found here. So those are the two distributions. So this is the distribution at 24 hours. So the probability of benefit was actually 94%, right? So that means the A 94% was better than the treatment B. Even though the relative risk confidence interval crossed the one, you have 94% of a potential benefit. So then then here's the question. Based on the Bayesian, you will say this was a positive trial, but based on the frequentist, you will say that it was marginal, right? So then what we can do in this scenario, right? That's the problem. The problem. This is actually a paper of 1 month ago. So actually this is ongoing discussion, because when this happened, the Bayesian is not pre-specified. So then how you justify which one would be your main decision? You see? So at the end, one of the conclusions is like it's in cases that you are pragmatic, right? It's better to have both, because in a pragmatic scenario, sometimes or in a very complicated disease where a benefit that is not statistically significant but is marginal is better than nothing, and it's not, it's safe and it's not so expensive, you could use it. So that's why having the probability is also helpful for decision making. Because in this scenario, even though with that non-positive trial, the people is still using this treatment A, you see, without the Bayesian analysis. But now with this, they try to justify why it works, even though the trial was negative.
Yeah, and that's the thing, I mean, exactly. You can't, and you're actually honest, you can't, if you want to go to Bayesian in the beginning, but that's exactly as Kevin was saying, so get some trials of the approval, but with actually start, you can't instead of frequentist and then let me see if it works with that's a no-no. But yeah, so I think that's the point is exactly seeing this, and then but also here was no prior knowledge, right?
Yeah, no prior knowledge.
Yeah. And I think the interesting is that to see trials. I don't know if you have an example of trials that use prior knowledge,
It's not common.
Not common, right?
No, because then it's as a sensitivity analysis, yes, but not as a main analysis.
Okay.
The main analysis is always non-informative or weakly informative. You can inform like you expect that your posterior will be normal, and that your mean would be in plausible ranges. Like you can have negatives because the biomarker can get negative values. You see, that is like weakly informative, but not with a point estimate that you expect. But you could.
But yeah, exactly. That would be interesting.
I know we cannot speak about alpha here, but if you could, it still would be a 0.06, right?
Yeah, it won't be significant if you use the alpha.
Anyways.
Yeah, and that's why at the end it's not the same. There is no correspondence.
What if this was like 98%? Would be more challenging.
Yeah, but usually if it's 99, the
then
frequency
will be
significant.
Oh, okay.
Yeah, the problem is this margin of probabilities of 80
very close
to 90 something.
Yeah.
Which clinically could be important.
Yeah.
Especially if you don't have any other option, if the treatment is safe. You see, so but
because in this case it will be as Kevin showed, be strong, correct? In your definition.
Yeah, exactly. It will be strong.
But it's not the 99% is the
yeah, let me show you
show the
optimization, because I think that's one of the important things to understand these trials.
Because that's it. So Bayesian analysis is more qualitative.
Actually, it's a positive.
Frequentist is what?
It will be positive.
Yeah,
positive, not even though it's positive.
But positive for Bayesian is considered a positive trial.
Yeah. But you see, but it's qualitative. Remember, is there going to be or now? P-value is categorical, yes or no. Here you have it 4 different classifications, and that's an interesting thing. So, but how to adapt that? How to then use that to clinical? Because we are frequentists, yes or no, and what's yes, we use in the clinic. But how to use a positive evidence, a strong evidence? Yeah, it is decisive.
Yeah,
decisive. That's fine.
Both sides will say the same. Yeah. But the other thing that they were also discussing in the paper is like at the end, it's kind of true that we are always trying to prescribe things that are highly strong evidence. But it's also true, like a lot of things that we are doing in clinic are actually not significant, but we don't have trials on that, right? So that's why actually it's even worse to prescribe that they are not significant, or at least if they are not significant in frequencies, they should have positive probabilities. Sure. You see, so that's trying to improve this analysis of the evidence-based recommendations, because sometimes, because of the type of data, because if it's pragmatic, external validity and issues, you just can get the
because and that's a big criticism frequentists, because so at the end of the day, you put the same bag is 0.06 p-value with a 0.99 p-value. You see, which are very, very different. 0.06, almost there. Likely, if you include 10 more patients, you will make it. But 0.99, you can include the whole population that you're not going to get it. So, and I think that's
another thing that at the end complements, as I mentioned, the interpretation of that. And then, but then it's not common to include very informative scenarios, but it could be possible. And that's exactly what I want to mention at the end. Like what we are doing now is translating this evidence to the generation to the application, right? So one alternative, that's the thing we are doing now with these models of machine learning, and actually Bayesian is really critical for machine learning, is to do this sequential learning, meaning the posteriors of now will be the priors of tomorrow, right? Because at the end, you are basing your analysis on your previous evidence, and we are always doing that. But now this is a way to integrate in the formal analytical pipeline. So at the end, what we can do is to accelerate this translation using this Bayesian sequential learning, and it's really it's really huge now. For instance, all ChatGPT is using this approach. When you text, when you enter a prompt, right? They are always using this prior assumption about you to improve the next text. That's why at the end, every chat that you have in this system is an example of Bayesian sequential learning, and it's using the same idea. So it's almost everywhere, except in the clinical care decisions. Right? We're always frequentist.
Exactly. That's and that's exactly, and that's the point that you guys need to think as well, how that would change, and the whole, I think, topic that I see that I think is very interesting is what we try to do in PPCR is to increase the critical thinking, because again, you finish medical school and you have no idea what is a p-value, to learn, but you don't have the conversation you have here. Maybe 0.0001% of the clinicians who know in terms of even knowing a little bit more p-value frequentist and what does it mean as well. And I think that's a very important thing, and how to translate that. And so because that's one thing. So this morning I was having the meeting with the he's a colleague of mine, coincidentally. So he moved to Switzerland and he works in Abbott, Switzerland. And then he contacted me. He wants to do a big program about Switzerland with Harvard. And we're just having this meeting with everyone. And the problem is exactly to discuss that this medicine, but in the context that it's not one size fits all. So we have what is the hormonal replacement. Before it was given to women, and then the evidence show can increase cancer, can increase cardiovascular disease, but still there's a percentage of women that does respond to hormonal replacement therapy, and they're not giving because we're just using this one size fits all. But that's the granularity that we need to understand, to practice it, and that's again the evidence-based medicine with critical thinking, or 2.0, whatever people like to use this term. And that's what we wanted to do with Emiliano, as Dr. Kevin as well, the precision neuroscience, and that's your thesis as well. So that part of your thesis. So using the biomarkers, so doing the whole concept of and be more critical and understanding more, and understanding the statistics as well more, and how to translate that.
Yeah, that's true. And at the end, I think we should try to integrate this more in our trials.
Yes.
And our analysis, because another concept is like, this is kind of a parallel world. So all the things that you could do it in the Bayesian. You could do Bayesian meta-analysis. You could do Bayesian sample size calculation, Bayesian t-tests, Bayesian regression. You just put Bayesian before and you can do it. It's true, because it's the same idea. So and we have the data to start with.
I think so.
Learning about it. And
that's why as well, so Dr. Kevin, that's why I think we need to, unfortunately, we need to move to R, because on everything in this data. And that was talking to Kevin, Dr. Alm as well, moving to R now or next year. So but again, because this tools is only R, right? Or Python, as you said. Maybe Stata will catch up in the future, but
It's true.
And also, Luan asked for these lights.
R is free. Sure, sure.
And I see, that's true.
Yeah, exactly. It's free. I think that's the beauty as well. Any questions? What's this?
Perfect, thanks.
Yeah, this is also, so any questions? Yeah. Let me just any questions on here, everyone. Let me stop the live stream as well. Once.